residues in different parts of chain. So we'll see that we have majorly 5 stabilizing interactions: hydrophilic, hydrophobic, then we have disulfide and also hydrogen bonding, also salt bridge. Okay, so first, hydrophilic interaction. So hydrophilic interaction, as you know, in a protein molecule, the exterior is hydrophilic and interior is hydrophobic. So it is the interaction. Hydrophilic interaction is the interaction with the exterior aqueous environment and a polar amino acid residue, pulling them close to outer surface of protein, pulling them to outer surface of protein. We can take the example of, say, very simple example is glutamic acid. So what is glutamic acid? It is our CH with the COH and NH2 as in every amino acid. Then we have a CH2 COOH. Okay, so that OH and the outer aqueous medium like water will be interacting, and this will be pulling towards to the exterior of protein. Okay, exterior or outer surface of protein. Then we have hydrophobic interactions. A hydrophobic interaction means it forms the interior of the protein between amino acids, between non-polar residues of amino acids. These form a non-polar center at the interior of the protein. So example, take any non-polar amino acids. We have many, right? So we in this example I have taken this phenylalanine, that is CH2 with the phenyl group, and leucine. Leucine is nothing but CH2CH3 twice, right? Yes, so this both are hydrophobic. So this form a hydrophobic interaction. Polar residues of amino acids. It is the interaction between non-polar residues of amino acid forming non-polar center at the interior of the protein. Then we have salt bridge. Salt bridge is the ionic interaction between charges of acidic and basic residues of amino acids. So if yes, we can generate charge by removing or adding something to a amino acid. So here I have taken the example of asparagine and lysine. Asparagine is CH2COH, in which I have abstracted a proton to create a base. Then lysine I have protonated. Okay, so it is the ionic interaction between acidic and a basic residue of amino acid. So simple example is this asparagine and lysine. Lysine is CH2,4NH2. So I have protonated this and formed this. Then we have hydrogen bonds. Hydrogen bonds formed between H of polar residue and O or N of second polar residue. It is basically the hydrogen bonding we have seen in case of cartilages and all. So between, say, serine and so I have taken here arginine. Okay, serine and arginine. So it is CH2COOHNH2 COONH2. Actually this is, yeah, CH2. But ഒരു CH2 ഉള്ളൂ. ഒരു CH2 കൂടെ വന്നാൽ അത് ഗ്ലൂട്ടാമിൻ ആയനെ. So here we have only one CH2. So it is hydrogen bond formed between H of one and H of one polar amino acid and oxygen or nitrogen of another polar amino acid residue. Then we have disulfide bonds. Disulfide cysteine, SCH2SH, right? So cysteine, so cysteine is it is SH. So we should oxidize that to get a S. So covalent bond formed when SH group of two cysteine residues are oxidized, that is H is removed. Okay, so these are the major fine stabilizing interactions of tertiary structure: hydrophilic, hydrophobic, salt bridge, ionic interaction between acidic and the basic residue protein residues. Then we have hydrogen bonds. As you know, it is between a H of a polar amino acid with another or nitrogen of another polar amino acid residue. Then we have disulfide. It is a covalent bond that forms between SH group between two residue of two cysteine residues are oxidized. Okay, so this is it. Now the interaction between amino acid